Cool. So this is where we left off yesterday in terms of talking about partitioning heritability. So we're just going to pick up there. We're through most of the math. There'll be a little more math when we get back into genetic correlation, but we're just going to cap off, remind what the idea was here on partitioned heritability, and then spend most of our time talking about kind of applications of both this and genetic correlation in terms of looking at results and thinking about kind of interpretations for these things. But to kind of reorient from where we left off when we were talking before, so you'll recall that that we kind of worked through an expectation for our GWAS results, so particularly our GWAS betas and the resulting chi-square statistic, based on a model where instead of there just being one amount of variation in effect size for those betas, for the effect size betas genome-wide, that instead we're now allowing it to vary with some annotation. And so in particular had worked through to what becomes evident here, that when we're looking at SNP-J, we have our null expectation and our expectation that comes from confounding, so that we had this S sub j here, that is now just that variation of that term. But we also now have that you sum across annotations and across all SNPs, looking at SNP-J's correlation with SNP-K, so we're indexing the other SNP by K here. So we have that squared correlation. And then we have the annotation for SNP-K for the category that we're looking at. We'll sum over all categories, or all of our annotations. And our effect size in that category, which is these taus d. So the intuition here is that it doesn't matter directly to the model what the annotation is maybe for SNP-J, but it does matter what the annotation is for all the SNPs it's correlated with. So even if you're not as a SNP in the coding region, if you're in LD with something that's in the coding region, then we might expect you to have stronger effect sizes if there are stronger effect sizes in the coding region, because you're still tagging that information even though the annotation doesn't apply to you directly as a SNP. And so off that model, we then have these LD scores that are for each of those dimensions that consist of summing over that annotation, that for the LD score for SNP-J, for a particular annotation, is the sum across all the SNPs of the LD to that SNP and the annotation of that SNP. And so kind of where that leaves us, that's both here and will become the top of the next slide, if I actually hit the button. Or maybe not if I've lost focus, which I have. There we go. So where that leaves us is with now, instead of the simple linear regression we did in talking about LD score yesterday, we now have a multiple regression because we have multiple of these LD scores and each one having a corresponding tau. And so, but as we were able to do in kind of the univariate version, we can estimate these LD scores from external reference data and plug this in along with our GWAS chi-square statistics and fit this model to get estimates of what those taus are. And those taus being, whereas before we had a single parameter that was something like SNP heritability per SNP, genome-wide, now we have a contribution of SNPs for a particular annotation in the same kind of scale of variance of SNP heritability per SNP. And so in application, we end up with many possible things we might want to plug in here in terms of our annotations. So the top table here with the header in blue is the set of annotations that was looked at in the original paper that Hillary led. Largely oriented around functional genomics categories, so from kind of the molecular genetics work that Andrea talked about a lot earlier this week. So things like this idea of what regions are coding, what regions maybe have elements that are known to not necessarily code directly, but have some regulatory function that affect the levels of transcription and such. Also some thinking about kind of more from a population genetic standpoint, regions of the genome that are maybe conserved, and so not even necessarily conserved across human populations, but really conserved going back through, say, all of mammals. Regions of the genome where the sequence has remained highly stable through that millions of years of evolutionary process, and thus maybe as evidenced by them having remained stable, maybe there's more problems or more likelihood of causing problems if you mutate in those regions, and thus maybe, at least in some circumstances, things will have larger effect if you have a variant that is in those regions. And so that was kind of one set of things, and the follow-up that happened in that initial paper was, well, so we can take that as our baseline about what we know about the genome, but then maybe also layer on top of that our knowledge about particular sets of genes that might relate to tissue or cell type that might then inform where, like, relative to like the general functional landscape of the genome, can we start looking at the GWAS and saying things about enrichment in a tissue or a cell type or a particular pathway or these kind of things that might go beyond just saying, well, yeah, things that are coding matter, but genes do lots of things, so maybe we want to get to narrower sets that are informative in some of these other dimensions. And so the kind of other thing that got considered in the initial set, which is part of what's being bullet pointed here, is that maybe in defining these annotations, we may want to consider also the regions like directly adjacent to these regions. So this is probably most clear if we're again thinking about something like the coding regions, where especially, so we specified our model that what matters is your LD to a SNP that's in an annotation. But it might be that, like, even if the SNP isn't quite in the coding region, so it's not in the exon of the body of the gene, maybe if it's like right near it, there's more likelihood that it has an effect. So maybe a variant there is likely to throw off the transcriptional machinery because you mess with the reading the start site or a stop site, or you mess with splicing, or you affect just how available the DNA is for transcription, all these kinds of more cryptic things. So maybe in addition to modeling each of these regions as an annotation, we also want to include on top of that an annotation that essentially is that known genomic annotation plus some buffer around it. And this does indeed, in terms of fitting the model, seem to make things behave a little more nicely if you add something like a 500 base pair buffer on either side. So that was the set of considerations in the first paper, and then this second table here that's below the bluer table is a set of continuous annotations that got added from starting at that paper and going forward to the Stephen Ghazal paper that generalized to continuous annotations. And so there was one other paper in between those two, this Liu et al. Paper. But so the idea here was that what things could be added once we had the flexibility to include continuous annotations. And so the main thrust of Stephen's paper was specifically to look at annotations largely related to more population genetics forces. So things like GERP score, which is intended to be another measure of likelihood of there being a functional consequence of a particular variant. Things like background annotation statistics. This is a statistic that tries to measure, again, kind of the selective forces that may have been acting in that region of the genome over a particular range of history, so operates on kind of a different time scale maybe than conservation back to all mammals, but is again informing you about those selective forces. You also have things like CPG content and nucleotide diversity that get at kind of this idea that there are some regions of the genome where it's just long repeats of the same thing and looks like it's space filler, versus other places where there's more variation, and so maybe it's encoding something more meaningful, so maybe that matters. And CPGs being particularly relevant because those, as a base pair together, C to G in order along the genome, tends to be more mutable in the human genome. So this has been one of the observations out of the NOMAD project that Alicia brought up, which is that if we look at across the spectrum of variation in whole genome sequencing and just kind of break down the mutations in terms of what the reference allele was and what got changed to, these CG sites are much more frequently mutated in the human population than any other base pair context in the genome. And so maybe that means that there's something informative there, and so maybe we want to model that and that there's something there in terms of thinking about effect sizes as well. So many different things we could look at. And so with all those things in mind, we can also think about like what metric do we want to evaluate these annotations on. So we fit these taus, and these taus are all in this conditional multiple regression model. And so kind of perhaps out of order here, the second bullet point is that we might just want to look at those taus themselves. And so the idea being that the tau for a particular annotation tells you about the added per SNP variance in the effect sizes, conditional, and all the other annotations in the model. And this makes sense, especially in looking at the things like the cell type and tissue type enrichments, where we would want to condition on the, like, you know, there's all these places that are coding. We know probably the coding things are enriched already, but are you enriched beyond what you would expect from the coding annotation? And so if we're interested in that, then we can just look at these taus directly. And perhaps if we're going to do comparisons across different traits, standardize these taus to the total heritability of the trait so that they're on kind of against a baseline of total H-square SNP. The other thing we can do, though, is what's in this top equation, is that instead of looking at the individual annotation by itself, conditional and everything, we make some statement about the total SNP heritability that's in that annotation relative to genome-wide. And so this is the one equation that I didn't manage to get in a slide, but thus put here, which the idea being that in a statement that only makes sense really for our binary annotations, where either you're in an annotation or not in an annotation. But so for things where we have a categorical annotation C, we can say that the SNP heritability for that category C is if we sum over all of the SNPs that are in that category, so not all SNPs, just the SNPs that are annotated in that category now, and sum their effect size from now all of the annotations based on their annotation and their tau. So it's implicitly here, then, we're taking into account the overlap with all the other annotations. And so, you know, maybe we fit a tau that says that you know, these conserved regions are really important, and so the conserved regions deserve a big tau. And so, and a lot of the, say we're looking at coding SNPs here, so a lot of the coding regions are conserved maybe. So, you know, we end up with lots of places where there's overlap. But so then when we look at the total SNP heritability for the coding category, we'll want to incorporate the effect that is modeled by that tau for the conserved category. And so we sum over all those annotations for the SNPs that are in that category to get this quantity. And that's the quantity that then plugs into this sense of measuring enrichment, that we're then taking that heritability per SNP in category C, so that MFC is just the number of SNPs that you had here that are elements of that category relative to the total genome-wide SNP heritability per SNP. Patrick.
So this is something I hadn't realized, summing over the categories. So if we didn't sum over the categories and we just stayed within like the annotation that we're interested in, then that would be interpreted as like the amount of heritability in the SNPs in that category due to the particular annotation, right? But that's not what we want. We just want to look at all of the SNPs in the categories and the heritability due to all the reasons, right? So
yeah, so there are circumstances where we may be interested in either quantity. And so part of the idea is something like enrichment, where thus you're summing over categories, is hopefully stable-ish, regardless of what exactly you've put into like your joint fit of this multiple regression for the model, versus depending on what all is in your multiple regression, all the annotations you're fitting at once, those taus can move around a lot, especially if you have strong amounts of overlap and correlation between these annotations. And so this is very much about like conditional and everything else. How valuable is this particular annotation to modeling the per SNP heritability, versus this is about like total genome-wide enrichment. And we'll talk about some results from each of these that kind of highlight where maybe each of those things is useful to look at. Cool. Any other questions before I flip to the first result slide? Great. So results. So we'll start with that enrichment metric. And so this is the set of results out of the first partitioned heritability paper. And so what we have here is 5 of the different annotations that were looked at that were all functional genomics annotations. And the colored bars correspond to 5 different traits, so GWAS of each of these, except for immune traits. It's not a specific disease. It's a meta-analysis of things. But so generally finding that, so in terms of this overall enrichment, and maybe makes sense that like enrichment in coding, you don't need to, maybe it's interesting that you're enriched in coding regardless of whether you can maybe model finer pieces of that. And so you've got maybe between a 5 and 10-fold enrichment of heritability per SNP in these coding regions based on these GWAS relative to the genome-wide average. A much stronger enrichment for most of these things out of conserved regions, though the kind of quirk here is, since we're talking about enrichment, this may or may not give you a statement about like where the total heritability tends to lie from SNP heritability. So that becomes evident, for example, when we're looking at, say, the conserved SNPs, which are about 2.5% of the total common variance, where with the definition of conserved that was used for this annotation fit that definition. So the kind of statement about enrichment here is, so as if you're at this 10-fold enrichments, then that 2.5% of SNPs are explaining, say, 25% of the SNP heritability. But that means still 75% of the SNP heritability is somewhere else. It's not all in these annotations. It's just that this small group of SNPs is kind of outperforming expectation. This has become a big talking point in terms of the omnigenic model from the Pritchard group, that is kind of pointing out this observation that even when we have very strong enrichment for a category, that doesn't mean that like all of the heritabilities in that category, or even most of the heritabilities in that category, in terms of dividing up SNP heritability, but rather just relative to the number of SNPs, it's larger. Yeah.
So the conserved SNPs, I mean, by definition, wouldn't the allele frequencies be low for those? Because, I mean, wouldn't you expect that these would be rare variants? So how exactly would you, like, how would they show up in a GWAS study? How would they be powered?
Yeah, so for the definition that was used here, it was done in terms of the conservation of the region rather than basic pair resolution, so it did permit regions where there's still some common variance there. It's just that on average the region is more conserved. I don't know off the top of my head like what exactly the metric was that went into that construction. I think it's a Trinka paper, if I remember the first author correctly. But yeah, something along those lines as a regional annotation rather than per base pair. Yeah.
So this is super interesting. I was just wondering, like, could we use this type of information on GWAS, just be a little more informed about which SNPs we should expect going forward, or is this not a good idea? I don't know.
So it's definitely something that could be done, and I think there are some programs that in varying degrees try and do that. So you could try and include it as a prior in your GWAS. There are various methods that apply it more in polygenic scoring, that when you're doing the kind of Bayesian shrinkage on your effect sizes there, that maybe in addition to knowing the p-value of the SNP, you also prioritize a bit based on other annotations. The kind of key discussion point around that seems to be like deciding when you want to apply that information. It's like you can gain power for locus discovery by setting the prior that way, but then you can't like check downstream like what the enrichment is because you've already loaded that in your prior. And so making those decisions are complicated, but in the general sense, yes, there's certainly room to include information that might be informative if you want to trust it as a Bayesian prior. Cool. So that's categorical functional annotations. And then with the continuous annotations, it's a little harder to represent what that tau means because it's just a single value. But one way to do it is to think about dividing up that continuous value, into quintiles or some other quantization, and look at enrichment within a quintile, kind of implied by the coefficient tau on the continuous value. And so that's what's been done here. And there's a script that does this that accompanies the LD Score software. But so this finds that, for example, things, if we look at, say, predicted allele age, which is, again, trying to get at, like, in this evolutionary time, maybe more common things are older, and if you've been around for a long time, you aren't being selected against, versus newer things have had less time for selection to act on you. And we see that the enrichments here maybe fit with that. So alleles with lower predicted allele age have a higher proportion of the total SNP heritability than the variants that have the oldest predicted allele age. So maybe there is this kind of sense that effects are proportionately larger when there's been less time for selection maybe to act on you in terms of your current effect size. Similarly, things that are in regions with high nucleotide diversity have are having less room here to are contributing less versus low nucleotide diversity is contributing more to heritability. You know, reasons why that is are kind of complicated maybe. Makes more sense in terms of something like background selection, or again, higher selection, higher effect size, higher CPG content where things are more immutable, higher total contributions to heritability. So places where, so again, just a sense of getting a feel for what these effects look like and kind of the interpretation that comes out of being able to look at these partitioned results. We can also start thinking about this as at scale and in terms of the individual tau coefficients. So this is results from a few hundred traits out of UK Biobank where we've done partitioned heritability and just looking at the general trends in those results. And so what we see is strong signal of enrichment in many of the different categories out of this model, but the things that pop out most strongly and most reliably are these, the CPG content measure, this GERP score measure that has to do with just kind of predicted deleteriousness of a mutation at that site, and this conservation measure as things that are very consistently across many of the things measured in UK Biobank, kind of punching above their weight in terms of contribution to heritability, SNP heritability per variant. And so this is based on looking at the tau, so this is conditional on everything else. And so kind of the other place that we've looked more frequently at conditional effects is in getting into those annotations that are informative about like cell and tissue type. So this is what the initial analysis of this from the 2015 partition paper, where have taken chromatin information, which is those kind of annotations of not the DNA itself, but these kind of molecular marks, is particularly these histone marks that are attached to the chromatin, and kind of assessing their behavior in samples of different tissues as potential marks of where DNA is available for transcription or not, and turning that into an annotation about certain groups of tissues based on somewhat complicated hierarchical clustering that was done. But kind of at the end result of that with the annotations that were put together, what we have is that, for example, if we look at the schizophrenia GWAS, and we look at an annotation that is, again, a binary annotation of, is this a region of the genome that is generally, based on these histone marks, active in central nervous system? Then in schizophrenia, in this orange bar, we see a very strong signal in terms of the testing this tau, that conditional on everything else we know about the functional landscape, that indeed there does seem to be some additional contribution to heritability per SNP from the things that are in these regions of the genome that have these chromatin marks, specifically in central nervous system tissues. And we see like similar signal of central nervous system in bipolar and in years of education and even in smoking. BMI as well is perhaps a little less expected. But that we can see versus other things where maybe we don't expect the central nervous system to have an impact, we can instead see contributions from, for example, immune system in rheumatoid arthritis, in Crohn's disease, is maybe that being more where the relevant tissue where it matters to heritability per SNP, that's where the signal is coming from. There's some other things here where there wasn't that much power for signal. The other one where there was definite power was in height, where we see a strong signal from, again, SNPs that are in these regions where the chromatin suggests it's active within, say, bone or connective tissue. Again, fairly face valid, and so a sense that the results we see make sense. And so kind of the follow-up on this has been that we can actually go beyond looking at just the tissue and get, as we've moved towards the availability of expression data, we can look at expression specific to the tissue and look at kind of more fine-grained things within tissue. So for example, we have a bunch of dots now that are all from CNS, and this is because we're looking at, say, different regions of the brain, different cell types within the brain, based on expression data from these tissues in some of these large efforts to put together references of what the transcriptional landscape looks like in each of these tissues. And so the model here was to take that gene expression data where there's been a test of the expression of a given gene in each tissue compared to all other available tissues, take the set of genes that is has the strongest signal for being expressed in that tissue versus all of the tissues. That gives you a set of genes to work with, and use that set of genes then to define your annotation for LD score regression. And so again, you're getting resolution about different tissues in schizophrenia, different parts of the CNS system, I should say, in schizophrenia, different parts of the system in BMI. And if we start to zoom in on what those things look like, we actually get into refining the tissue types. And so kind of the first part of this panel in block A is doing a comparison of within the brain tissues, is there a different signal for tissues, for expression that was tested in cortex versus cerebellum versus the striatum. So kind of a regional part of the brain type assessment. And so seeing there that, for example, in schizophrenia, there's a fairly strong signal suggesting that what matters there is maybe cortex as compared to cerebellum or striatum. Probably pretty consistent with like higher order functioning things. But interestingly, that there wasn't a similar association there in user education. Instead, that pointed to cerebellum. And that actually was the motivation to do follow-up, where instead of looking at regions of the brain, to look at specific cell types. So looking at astrocytes, which are kind of a structural manager in the brain, versus neurons that are actually the cell that has the synapses and is doing that connectivity. And finding that there you get the strong signal in user education that comes from neuron and is consistent with, might make a little more sense in terms of transmitting information, and actually kind of back tells us that perhaps what the signal that is being seen in terms of region of the brain is that it turns out there's in terms of the composition of the tissue, there's more neurons more densely in the cerebellum than there are in the cortex. And so what we're actually seeing in tissue type is maybe informative about cell type and hiding that. And so the importance and the information that comes from being able to look at these different enrichments through these different lenses. And then there is a little bit of work in that paper to even break down amongst the neurons, kind of what kind of receptor those neurons have between GABAergic and glutaminergic, particularly because there are hypotheses about this in schizophrenia, but is probably an area where the signal is a bit weak at the moment and is subject to, again, continuing to let the wet lab flesh out the data that goes into these annotations to kind of clarify that signal. Yes?
So it seems like these sorts of approaches are going to be sensitive to the annotations that you include. So like,
Absolutely.
You said that in like panel A we have cortex, cerebellum, all these things, but then in B we have cell types. But if we included like all 7, like what would happen in a case like on one hand maybe it'd be fine because if we're doing this thing maybe it all works out in the end. But did you have a sense of why you wouldn't just include all of these annotations at once?
Yeah, so part of it is the sense of the degree of correlation and overlap. That you're right that like you'll kill your ability to detect the signal to some degree once like all the annotations are very slightly modified versions of each other, and also just from an interpretational standpoint, like you may have power to detect that like you're enriched in neurons beyond an expectation, conditional on knowing that you're enriched in cerebellum and vice versa, but like the power statement required for that is probably worse than the power statement to just test it individually and be informative there. But you're certainly right that like given infinite power and enough room to differentiate those hypotheses, you could jointly model them. Yeah.
Where's the power coming here? Is like the number of SNPs you tested in the GWAS, or like what?
Yeah, so most of the power here lives in the sample size of the GWAS and the degree of SNP heritability. That's kind of your baseline that you're partitioning and breaking up into pieces. and then power relative to that based on the size of the annotation. So it's easier to say that, like, out of your million SNPs or whatever, that this 500,000 is doing more than the other 500,000, versus saying that this 100 SNPs is doing more than the other 99,900 SNPs. And to some degree, the model starts to fall apart once you try and get down to annotations that are that small. But so, like, relative to that baseline comparison of power for different annotations, the big drivers are the kind of degree of polygenic signal that was existing for your overall SNP heritability that you're then trying to pull into pieces. Yeah.
So this is a very basic question. I'm sure you've covered it. But so just a question about the annotation itself. So these are SNPs that are within genes expressed in certain tissues, or these are genes that are associated with expression of those within those tissues?
Yeah, so the annotation is based on the gene. So you define your set of genes that are in the annotation for this. And then that gets mapped to, here are the set of SNPs that are in that gene, that thus in terms of what actually gets plugged in to the LD score regression in terms of these A's is just an indicator of whether or not that SNP is in that gene.
And gene defined by gene boundary or plus minus 10?
I'd have to go back and double check which way it ended up being done here. I think they tried various things and they were all fairly similar, but I think it's probably gene boundary and a small buffer. Cool. The other thing that's relevant, especially since we've had previous conversation about the intercept, is that while most of the focus in looking at the partitioned heritability is about kind of looking at these enrichments or these conditional effects in the taus, but we do still get out of the regression both an intercept and a overall estimate of what the total SNP heritability is. And so looking at the impact of the partitioning compared to the kind of univariate estimate on that front. And so what we've observed from, again, running this in many traits in UK Biobank, is that when you do this more thorough modeling of allowing annotations to vary, you do end up with an estimate that's maybe ever so slightly higher for the total SNP heritability, and a corresponding slightly lower estimate on what the intercept is. That is consistent with an idea that while the intercept does capture confounding, it's also going to some degree capture model misspecification. So if you've modeled so you've modeled LD in terms of that you're expecting this relationship between the degree of signal in the GWAS and the strength of LD for a SNP. But if there's strong variation across different regions in the genome in terms of heritability per SNP, then essentially we've misspecified that univariate model. And so there's not going to be as much correlation between those LD scores and the GWAS results, and thus kind of the remaining inflation in the chi-square statistics that we haven't modeled that may still be polygenic effects but doesn't track with that single LD score will have gone into our intercept and will perhaps be modeled out a little bit by doing the partitioning. And so this becomes a little bit of a wrinkle in thinking about, like, now which intercept do we want to use for correcting our results for if we're doing inflating the standard errors out of GWAS. The reassuring thing is that they very strongly track with each other, so we're not making a huge impact regardless of whether or not we've done this partitioning. But there may be a suggestion here that if you correct from the univariate result, you're maybe ever so slightly overcorrecting. Though, especially in this region, you have to be at fairly high power for this intercept. There has to be a fairly strong intercept to begin with to be seeing these differences. The other aside that's noteworthy here is just in terms of that this is the distribution of the intercepts from estimating LD score regression for hundreds of traits in UK Biobank, where we've done the GWAS just in European ancestry individuals, and where we've included 20 PCs that were computed in UK Biobank. So they should be really good PCs. This should be a fairly homogeneous sample. But there are some traits where that's not enough. You still have fairly strong stratification here, and even kind of on average, you have a bit of a tail. You're drifting away from like having perfect control of stratification, this intercept of 1 that you might expect. Some of that may be remaining model misspecification, that there's other things we could annotate, but some of that is probably just that PCs aren't perfect at controlling for stratification, and we do see signs of that here. Cool. So last few notes on partitioned results. There's code and LD scores that have been publicly downloadable and are available. And I think we've already noted the point about the need for large GWAS sample sizes to be able to get reliable estimates out of LD score. That requirement only gets larger the narrower the partitioning annotation you want to look at, or the more things you want to include in your model. In particular, it then matters relative to the size of the annotation, and that in thinking about the LD score model, maybe the model starts to get a little unstable once you're wanting to model really tightly packed annotations. So we find the kind of rule of thumb, maybe 1% of the genome. If you're going less than that, things start to become a problem. And a lot of that problem lives around that we have to rely on these jackknife standard errors. And so if your jackknife only has information about your annotation is all in 1 block of the genome, and so all of your standard error that's being estimated by this block jackknife is based on the impact of adding or subtracting that 1 block of the genome, you're maybe not fully capturing the amount of variability you should, and so results get kind of unstable for those small, tightly packed annotations. The other thing that can be problematic is we've been careful in talking about these annotations based on genes, that it's a test of expression at the gene level, and we annotated the gene, rather than taking something like results from GTEx and looking for specific EQTLs and modeling those EQTLs. And so part of the reason for that is that once you get into defining an annotation based on the SNPs results, then now we've gotten circular with our expectation about how the SNPs behave. So if you've shown up in a GWAS, we already have a model that says you probably have a high LD score, and you're probably in an enriched region, and you're probably common because, power considerations. And so the SNPs in the annotation then are no longer have the same sense of randomness in terms of having a distribution of effect sizes. We've made this strong ascertainment out in the tails. And so, in times that this has been looked at, either intentionally or unintentionally, you just get really weird looking enrichments where, 3 times your total SNP heritability is in your annotation because your annotation is the SNPs that were the hits in the schizophrenia GWAS. And so that's kind of one wrinkle of there's a fair amount of flexibility as far as what annotations you can look at, but annotations that are circular with the GWAS are kind of a problem. The other thing that is sensitive in terms of thinking about those enrichments is that it is enrichment relative to a model of what variation is expected in a per SNP heritability kind of sense. And so you could make a different set of assumptions about how effect sizes should scale, especially with respect to LD, which are the set of assumptions that exist out of the kind of line of literature around LDAC and some HER that would suggest that your enrichment statement is a very different statement. And so if you compare these methods, you get really different looking enrichment results a decent proportion of the time. So there's a preprint that I think is optional on your reading list from work from Stephen Gazal and Hillary to kind of put these two models together, the LD score model of enrichment and the LDAC model of enrichment, and kind of model them jointly and look at what that looks like. But it's just a useful thing to think about, that there is kind of this underlying statement about when you talk about enrichment, what are you enriched compared to? Cool. That's the end of the partitioned heritability that we started yesterday, and then we'll get into genetic correlation. Any questions before we move on? Yeah.
So one of the categories that you had was GC content.
Mm-hmm.
Is there a bias in terms of SNP detection in higher GC content regions?
I am not necessarily the right person for that question. I know maybe less certainly about SNP detection, because we at least to some degree trust our chip design, but perhaps more in terms of that GC content relates to density of genes and other transcriptional machinery, and so is a useful tag on that kind of variation. I don't know if anybody else has a more useful comment. I'm getting a thumbs up, so apparently that's all I need to say. Yeah.
So you're saying annotations of less than 1% of the genomes is not recommended, but I mean, aren't most of the annotations that you showed us genetic? I mean, within genes, and that would probably be around 1%. So am I misunderstanding?
So down to 1% is fine. The lumpiness is in terms so you can definitely be like, here's a clump here, here's a clump here, there's a clump over there. The key is that you want cumulative 1%, and that you want those pieces spread out. So you don't want your 1% to be just that you've taken the first 1% of the genome and there it all is on chromosome 1 and there's nothing in the rest of the genome. That becomes more problematic. You want it dispersed across the blocks that get used for the block jackknife.
And one last. Sorry, go ahead.
I was thinking that's just for the standard errors, though, right? Like,
Yeah.
which is important, but yeah.
And then you had made this point that there's a circularity of using annotations based on GWASs themselves. That's really interesting. I hadn't thought about that. But that's irrespective of the fact that these two GWASs are completely independent of each other. It's just that GWASs tend to find similar types of things.
Yeah.
And so, okay.
Yeah, it's essentially your annotation is now loaded with a bunch of other statements about the SNP that aren't just its degree of effect size, because it has essentially a winner's curse type argument. So like if you had the infinite sample size GWAS and annotate based on that, you'd probably be fine. But relative to finite sample, now you have all these cryptic confounding considerations as far as what's gone into the annotation. Cool. So genetic correlation, which is the third paper off this list. Just in the past week, there's been a really, really nice review paper out in Nature Review Genetics that talks about not only LD score genetic correlation, but just the concept of genetic correlation more generally. There's kind of useful walkthrough from a conceptual side and kind of the uses and misuses of understanding genetic correlation.
Both of the first two authors are both named Wooter, which is a name I've never seen before.
Voucher is a reduction.
Oh, Voucher. That sounds like more.
Yeah. It's, just in the past week, so it didn't make the reading list for that reason. But so motivation here in terms of genetic correlation, now we're thinking about more than one trait at a time. But the idea that we've done lots of GWAS now on lots of different traits, lots of those traits are correlated, and so we'd be interested in saying something about the genetics of these traits that we know that are correlated, at least phenotypically. And so this isn't a new idea. You can go back to the example here is the first cross-disorder paper from the Psychiatric Genomics Consortium, where they looked at across, here's the 5 psychiatric disorders, so ADHD, autism, bipolar, major depression, and schizophrenia, and essentially went looking for SNPs that appeared associated with all of those disorders, and found in this paper a few hits, and in subsequent efforts are finding more. But so pointing out that, so, we have a variant here that effect size appears away from 0 for association with schizophrenia, and also for, what's the order here, depression, and for all the other things. And so this observation that in traits that maybe have some phenotypic similarity, so kind of in the domain of psychiatric disorders and kind of known comorbidities across these different disorders, that there seem to be some sharing of the genetic signal, as might have been predicted based on the phenotypic sharing, but being able to show that here. And it'd be useful, we'd like to be able to make some statements about the kind of degree of similarity across these phenotypes. And then both to talk about kind of the similarity in the genetics, just because we know it's partially genetics, partly environmental, so maybe talking about just the genetics is interesting. But also because in talking about just the genetics, maybe we either are able to make some statements that we wouldn't have been able to assess in the population, so in particular looking at like combinations of phenotypes that either aren't going to get studied together or are really hard to collect together. So you can imagine, wanting, your GWAS of some childhood onset neuropsychiatric disorder and dementia. You're gonna have to wait a really long time in that data collection, or really trust people's recall of their childhood onset disorder. And so, kind of the advantages of being able to do this from the genetic sense, where you don't need the same phenotype in same phenotype in the same individual, and wanting to be able to do this kind of in a genome-wide sense rather than looking at just the specific locus, whether it's shared. And so kind of going into thinking of a quantity of genetic correlation, we can suppose that we're back to our nice linear model. We're going to drop having the confounds list in the model just for simplicity, but now we have two traits. We have our y, we have our z, and I've adopted Patrick's notation from this morning where now we're for a given set of SNPs, and we now have the joint fit of those SNPs within. So rather than necessarily the true causal beta, this is like the best linear predictor beta. Like, given that I've picked my set of SNPs, as Patrick said, the data gods come down and give me the optimal set of weights for those SNPs to turn into prediction here. And so the idea is that having picked a set of SNPs, we might be interested in the correlation between the effect sizes that are here for this trait and here for this trait. Same sense that we talked about there being a universe of SNPs when we were talking about SNP heritability. And so to work through a definition of genetic correlation, there's a few different options. This is very nicely outlined in supplement EA paper, and I think is in that review. Not as much in that review paper as new, but is definitely in this EA supplement. So one thing we could think about is just the correlation to the effect sizes. So we have these conditional effect sizes. How much do they correlate with each other? But we might also be interested, and is actually perhaps the more common way genetic correlation gets talked about, in essentially the correlation of the total polygenic risk. So rather than worrying about, like, is the conditional effect at lined up on a SNP by SNP basis correlated, talk about the correlation of the total risk that allows for maybe it's not the same causal variant, but it's very much in the same region and trends in the same direction. And so once you account for LD, you'll see a lot of similarity. You'll expect a lot of similarity in terms of if somebody has a high genetic risk for schizophrenia, they also have a high genetic risk for bipolar, and maybe that's because they're shared loci, maybe there's not. We just have that observation, and we want to model that correlation. And so that's what this quantity is here. That kind of directly notes that what matters here is the LD matrix across SNPs becomes a feature here in this quantity along with the effect sizes. And we can compound this further and talk about the expected correlation of the marginal effects we see in GWAS. So remembering that our GWAS betas have to do with the LD matrix relative to these more causal or at least conditional effects. And so that essentially you'd end up needing additional weight on the LD. And this is perhaps more interesting if we wanted to think about how similar should the GWAS loci be when we look at the GWAS for this pairs of traits. And so each of these have their respective merits and are interesting in different ways. And under certain assumptions, they're equivalent. And the EA paper, I should be specifying EA2. No, these slides are from EA. From 2 years ago. Anyway, so the EA2 paper makes direct comparisons and finds that these, at least the first two quantities end up being fairly similar. And so the reason I kind of walk through that is partially because for LD scores purposes, we're going to estimate a quantity that is based on the correlation of effect sizes, but we're going to make some assumptions so that we can make the claim that that it is equivalently an estimate of this correlation of the total polygenic risk. So quickly, the layout for that model then is to say we have our two traits. We're going to say that the covariance of these set of effect sizes, so the variance within a set of effect sizes is proportional to this heritability per SNP. So we're normalizing per SNP out here. And we have different heritability for trait Y and trait Z. And then we have this covariance of those effect sizes that will turn into our parameter of interest. But the other thing that's lingering here in the specification here is that within each of these blocks of the matrix, these are diagonal matrices. So similar to the assumptions we made back in talking about a single trait, these are random effects that are uncorrelated across SNPs. So across the set of SNPs is why this I is a block. And so there's no correlation across the SNPs within trait. And when we're talking about the correlation across traits, that's correlation of SNP J in trait 1 and SNP J in trait 2. Those effects are correlated, and that's what we're modeling. There's no correlation between SNP J in trait 1 and SNP K in trait 2. All those other off-diagonal correlations are 0. Simpler modeling on the error term, that there's, the error variance is whatever the SNP heritability is not, and there's maybe some environmental component to the covariance. So that's a kind of implicit parameter here that is allowed for, and essentially assume that all the things that aren't otherwise specified up here as being correlated are conditionally independent of each other. So the effect sizes are independent of the errors are independent across individuals, other than the correlation between the error of the two traits for a given individual. And so I've set this all in terms of an individual, but we're going to be modeling in terms of effect sizes. So in some ways, the independence of the errors across individuals ends up being less worrisome, other than when we talk about sample overlap, which we're not talking about in too much detail. So we're going to focus instead on that we've stated this parameter, this rho sub g, that is a covariance term for the covariance of the effect sizes. And now we're going to make the justification for why we want to call that our genetic correlation that relates to the covariance of total genetic risk rather than just the correlation of the effect sizes. Yeah.
On the last slide, that's not a statement about, the multiplying for the identity matrix. It's not a statement about LD. It's a statement about the actual effect sizes being correlated. Right. Yeah. It's just these effect sizes. The LD still exists. And we'll see the LD propagate back into the model on the next slide, I think. Cool. So if we were thinking about the correlation, or we'll stick in terms of the covariance for the moment, the covariance of the polygenic risk, we can make the natural expansion that so each of these is just the sum over the effect size and the standardized genotype. And so that covariance should take the form of essentially the covariance across. So you have kind of covariance within trait in terms of this variability that the what is the term I want? Yeah, so you essentially have the variances and the cross terms in terms of the effect size. And so what we want to do is that essentially we're going to get rid of all the cross terms in terms of different subscripts that corresponds to the assumption that we just made back here, that this is an identity matrix along the diagonal. So as long as we can make that an assumption, then when this simplifies down to that you don't have to worry about cross terms between the SNPs, because again, the effect size of SNP J and SNP K is independent both within trait and across trait. So all you need is the sum of essentially the elements that exist at a single SNP. And so like that, as Matt already asked about, that LD still exists, but because of the random effects assumption, and that we're saying that within a trait, the causal effect of SNP J and K is independent, and across traits, the effect of SNP J and K are independent as long as it's separate SNPs. Then what we reduce down to is the statement about the variance of the SNP and the cross product of the two things. Since we've standardized our SNP, all we're down to then is the expected cross product of the two effect sizes. Since we've mean centered the effect sizes, that's then just the covariance of the effect sizes. And so this gets us from the parameterization we just made, where we have a covariance of those A's and B's, now to saying that covariance of A's and B's should also be our covariance of the total genetic risk accounting for LD, based on this assumption that we kind of random out the cross terms in terms of their correlation of effect sizes.
Should it be like an M, or because you're summing over a bunch of rho G's, right? Or are you defining rho G?
There should be an M. Yeah. This is yeah. There should be an M. Cool.
And it only works when X is standardized, right?
Yes. Yeah, this is all. So you can derive an equivalence if you want to plug in allele frequencies and make some statement there instead, but this is all living in the version where X is standardized. Cool. And so if we pick up from our previous derivations for LD score regression in the SNP heritability case and the partitioned heritability case, we again have this expectation about kind of the components in terms of the statement about what our GWAS estimates look like. And so what we're going to do is we're going to say we have these GWAS estimates. We now have the corresponding Z statistics for those, so the beta over the over the standard error. We're working in so 2 notes. Notationally, we have these as zetas to avoid confusion with the Z's that we've talked about as covariates elsewhere. Z's would be natural to talk about this, but we'll do zetas so that we aren't overlapping in that sense. And then we're now operating on the Z-scores instead of the chi-squares, so we're maintaining the sign of the effect. So we're no longer squaring this numerator so that we have, right, so the sign still matters. And so what we're going to do is we're going to just magically decide that the right thing, obviously, is to take the product of these z-scores and model that, and it'll turn out that it works. So if we look at the expectation of the product of these z-scores, you have, the top statement is just a restatement of what we just said these z-scores are on the previous slide. But then once we incorporate our assumptions, again, about the kind of independence across SNPs, they get rid of these cross terms, and we're also going to make an assumption that similarly the confounding in one of these traits is not correlated with the effect sizes or the LD or the error in the other traits. So we've already made this assumption kind of within trait that the confounding is independent of the LD and those effect sizes and of the error, but now we need to also get rid of the corresponding cross terms that exist here. So making that additional set of assumptions, then the math walks through, and what we end up with is this bottom statement where the expectation of this product of the z-scores is this equation that has to do with the magnitude of the confounding and the error, and the cross product of the error between the 2 sets of GWAS statistics, and this piece that is proportional to our target rho sub g and the same LD scores that we defined previously in terms of just a single set of LD scores. And so again, this gives us our nice set of regression. We have our z-scores from our GWAS. You take the product, you plug those in, you have your LD scores, and you now have a simple linear regression that has an intercept that is now this quantity and has a coefficient that is proportional to your rho sub g and your known m's. Cool, and then kind of to complete the circle, that gives you a covariance term. You do the SNP heritability estimation within each trait to get what the SNP heritability is. And so you can then turn the covariance into a correlation just based on normalizing for the 2 SNP heritabilities. Cool, so I kind of breezed past what that intercept is there. It's a big, ugly looking statement, but essentially it's just that it scales with now the combination of sample sizes and combines either a correlation of the confounding between the 2 traits that you're looking at or a correlation of the error terms. And so these are kind of 2 potential ways that the studies could overlap in a sense and look more similar than where the GWAS would look more similar than if we had purely independent studies that just are similar because the genetics are similar. And so in the sense of the error terms, the way we can end up with correlation of the error term between the 2 GWAS is in the simple case of sample overlap between the 2. So you have the same individuals who are studied both times, so even up to the level of that we did both of these GWASs in the same set of people. Kind of the extreme case. But the other option here is that maybe it's not the same set of individuals, but there's a similar pattern of the confounding. So we did our GWAS of height out of a broadly European sample once, and we found an entirely separate group of people, European ancestry, and did our GWAS of height again. And while height still correlates with the kind of same axis of genetic variation in both of those samples, and so we might then see shared confounding if that confounding for stratification hasn't been fully corrected out of both of those GWASs. So that will live in this intercept as well. If we assume that kind of overlap doesn't exist, then we can simplify down our statement about what this intercept should look like to just a statement about the number of overlapping individuals and the phenotypic correlation in kind of that simpler case to get a better intuition for how this quantity scales. But it does indeed capture both this combination of sample overlap and confounding overlap. So as has been in all the other sections, a few miscellaneous notes of kind more applied considerations. So basically all the statements we've made about the univariate model apply here, that we're still relying on block-Jackson standard errors. We're still waiting the regression for heteroscedasticity. The interpretation in terms of whatever statement about the correlation of these SNPs is relative to a selection of SNPs that we've included in the LD scores, and that like all of the LD score methods, if you had individual level data, or if you wanted to make maybe different model assumptions, there are alternatives and potentially more powerful alternatives. This is just a quick and easy way that has been useful so far to get at this quantity of genetic correlation. And so, a couple of those potential directions, one of those potential directions for thinking is that we've kind of implicitly assumed in working to that derivation that we have LD that's the same in both of the populations for our two traits. If that's not the case, then we've broken our model of the LD scores in ways that break the estimation fairly noticeably. And so there's a further development in this method called popcorn that essentially gets you cross-population LD scores that seems to maybe sort of work, but still be kind of rickety depending on the nature of your reference samples, but at least provides some options outside of LD score if you're interested in this quantity of cross-population cross-population genetic correlation. And perhaps one nice thing that is what's in one of the main outcomes of James's paper about LD score regression is that one of the nice things is, since we're looking at the correlation as a ratio between the covariance out of this regression and then getting SNP heritability is also at LD score, to some extent, the biases in LD score where you might be underestimating for any number of reasons, or you might be worried about various forms of biases. By looking at the relationship between these estimates, at least some of those biases cancel out. And so perhaps you're more robust in terms of statements about genetic correlation, and also in terms of less formally stated but similar intuition, that there may be similar robustness in looking at enrichment and partitioned SNP heritability than you are in trying to make statements about the raw magnitude of the SNP heritability, or if you were trying to make raw statements about the magnitude of the genetic covariance rather than the genetic correlation. Cool. So limitations. So power, power considerations are pretty good. This is the one place where we maybe have started to make quantitative statements. So since we're on block-Jackson standard errors, it's hard to make analytic statements. But you run this thing enough times with enough traits, maybe you observe how it behaves empirically. And so this is results from the Brainstorm consortium that looked at genetic correlation across a lot of psychiatric and behavioral and neurological traits. And so the orientation here is that we're looking at the square root of the product of the sample sizes and the SNP heritabilities as our rough metric as far as our intuition about what power in LD score regression depends on, against the inverse of the standard error here. So standard error, so at 10 here, we're looking at a standard error of 0.1, 20 is a standard error of 0.05, etc. And so the useful thing here is essentially, since this is the root of the product, if we fix, say, sample size and heritability the same as the two traits, we're saying that maybe at a heritability of 0.1 and a sample size of 50,000, that you might have a standard error of your RG somewhere between 0.1 and 0.05. Or if your heritability was 0.2, you can maybe accomplish that with a sample size of 25,000. But at least statements like this start to give us at least some intuition or quantitative statements about roughly what the power expectation is based on the behavior of the standard errors. We've started to make similar statements about heritability and partitioned heritability estimation as well. We just aren't as far along on that. But this is rough, and you can see that there's a spread here that probably at least partially is catching that idea that since we're in block-Jackson standard errors, not only does the heritability matter, but the kind of distribution of the heritability across the genome matters, kind of in the similar way where we're concerned about annotations that are really, really location-specific. If the polygenic signal is more location-specific, then we might see larger standard errors than if it was really, really flatly dispersed across the genome. Cool. So the talking about genetic correlation doesn't get you away from the nature of the phenotyping in the GWAS. And so perhaps the cleanest statement of this is if you look at the same phenotype or at least nominally the same phenotype, in different cohorts that are large and take the genetic correlation between those cohorts, it is not entirely unusual that you might end up seeing differences. So in some ways, this is a nice QC metric, but in some ways is also interesting in thinking about heterogeneity and meta-analysis and thinking about what it means, to observe kind of mid-range correlations. But so for the kind of main focus, this result out of the major depression group was so the PGC-29 is their meta-analysis of 29 research-collected individual cohorts from across the PGC. And then they've looked at genetic correlations here with 6 other larger, mostly more biobankish cohorts, and see kind of a range of correlations. Often the standard error is large, but at least in some instances where, say, for example, iPsych out of Denmark or data from 23andMe, you're seeing things where relative to these standard errors are probably significantly away from 1. And so there's some difference across the cohorts here where even though nominally all these cohorts are looking at major depression, they've asked for trying to collect the same phenotype. But the genetic correlation is exposing that there's cohort across the phenotypes. So this is both interesting in and of itself, but also something to think about in terms of when you interpret the genetic correlation, what it means that the correlation with depression may matter on what GWAS of depression that you're comparing against. And kind of last point here is that it is just a correlation. So all the kind of snafus of what a correlation means still apply, that it's not causation, implies maybe some similarity in terms of pleiotropy relative to how you've measured things, and there is at least a consistency of sign component. But we can imagine that like our mental model of the correlation is probably something like this. So like if we have our effect sizes from trait 1 and trait 2, a really high RG here looks something like this, and you've got this nice pretty regression, and maybe your ridge correlation looks like this. And if they're uncorrelated, then there's no correlation. But we could have the exact same correlation between those phenotypes if, for example, there was some stuff that was specific to trait 1 that's not part of trait 2, but the rest is perfectly correlated. And so you have this piece that's unique versus this piece that's shared, and that has exactly the same RG that this does. Similarly, as you go down, the statement ends up looking even worse. And actually, it's an interesting comparison at the uncorrelated end, because this actually exposes that while this said that our genetic correlation was 0 because there's no correlation to the effect sizes, this actually says that all the same SNPs are having an effect, it's just there's no correlation to the effect. Whereas here, it's an entirely disjoint set of SNPs. And so, something to keep in mind in terms of these quantities. This is a really simplified model. We don't even have a component here where the correlation's in the opposite direction, or there's unique effects of trait 2 that aren't part of trait 1. But so all this kind of loads into what we've stated as the single parameter of genetic correlation. There's some methods that try and parse out these pieces, but again, as you're trying to carve up pieces of the distribution, that gets harder than a statement about the overall correlation. Yeah. Here.
Are those offline on correlations? How come some are above 1?
Right. So LD score genetic correlation, because it's that slope out of a regression, and we're taking a slope relative to other things, isn't bounded in the way a classic correlation would be. All these are within standard error of 1 easily. So like if we start seeing correlations that are significantly greater than 1, then we probably have a problem. But it's not a bounded estimator, and in fact, we want it to be unbiased, so there is room for variation that when the true correlation is 1 or roughly 1, that you see estimates that are greater than 1. Cool. Anything else before we take a quick look at the nature of results and call it time?
Do people ever use within sample estimates of LD? Instead of reference time?
Yeah, so I hear conversation about it all the time. I don't see it hardly at all in the literature, and I don't know if that's because people decide it's not worthwhile or nobody actually follows through on the thought about the value of in-sample LD. The one potential quirk of in-sample LD is that, so at least with how we've approached the LD scores out of 1,000 genomes and such, is you have the advantage there that there's some underlying sequencing data. So you have unbiased capture of the SNPs across the genome, and thus your LD is capturing that full range of variation. versus if you wanted to do in sample LD, unless you have a sequence sample, and if you have a sequence sample, you probably aren't doing LD score genetic correlation. If you had, if you don't have a sequence sample, then either you're trying to compute LD scores off of imputed things, or you're restricting what set of SNPs that you're including in terms of your modeling there. But at least in principle, you're right, the in-sample LD would be ideal.
It's a trade-off, right? Like, so you can have in-sample LD but not sequenced, and so it's not quite right because you're missing stuff that you didn't genotype, but your LD patterns are going to be exactly right. And so, you just have to weigh that, I suppose.
Yeah. And you can imagine there'd be a sequence subset of, like in UK, that indeed this exactly happens. And so you think maybe you'd want to be using the 10,000 UK 10K sequence data for doing LD score regression.
Yeah, so I think UK 10K is like the one place where I've seen people actually actively looking at then it's like within sample subset LD scores, but at least putting more emphasis on the closeness of that versus 1,000 genomes.
There's no sort of worry about circularity or anything, or overfitting or any kind of issue like that there, because you're estimating two completely different things, right? The LD and then the other is the...
Yeah, you get overfitting if you were looking at like the relationship to the phenotype, but if you're just looking at SNP relationships, then...
Yeah. Cool. So we're going to just quickly breeze through an example of what these results look like. I realize we're pretty close to time. So this is the results of the first genetic correlation paper that kind of highlighted that within domains of phenotypes, you see a decent breadth of correlation across things. So in terms of, say, metabolic outcomes, body size, the psychiatric phenotypes, the combination of ulcerative colitis and Crohn's disease, like there's definitely groupings of things that might have been phenotypically expected to go together, and genetically they seem to track together as well. We saw some instances of things where there either wasn't a predicted correlation, or like even the sign of the correlation was different than expected. So one that's received a lot of attention that wasn't significant here but became significant in later analyses was a positive genetic correlation between autism and years of education, whereas the epidemiological literature and phenotypically there's a fairly strong negative correlation there, so it was unexpected to see a positive correlation here. On follow-up, it's kind of fairly strongly pointed to the distinction between a statement here about correlation out of common variants versus maybe the contribution of rare variants that are important in autism that have a detrimental effect on cognitive ability, and thus kind of phenotypically overcome the positive genetic correlation seen in the common variants. You also perhaps end up with interesting cases where you don't see a correlation, where you might expect one phenotypically. So in the case of schizophrenia, the smoking is near universal amongst individuals with schizophrenia, to a point that I've been told that it's nearly impossible to study smoking behavior amongst individuals with schizophrenia because everybody smokes, so there's no control group, basically. But in terms of the genetics, we don't see a genetic correlation between the GWAS of smoking behavior and the GWAS of schizophrenia, and so maybe that has something to do with what drives individuals with schizophrenia to smoke is different than what drives most of the general population. And if you haven't represented, that many individuals with schizophrenia in your GWAS of smoking behavior, then maybe you don't see a genetic correlation, and maybe that's noteworthy even though it's a null finding. These are the genetic correlations that were in the EA2 paper that are here because it didn't end up a main text figure to look at genetic correlations in EA3. There's a whole range of interesting genetic correlations with reproduction, also from the reproductive behavior, also from the SSGAC, that kind of implicates not only things that are more like directly around sexual reproduction, but also many things related to kind of behavior, and like years of education pops out strongly there. Newer set of results out of the risk-taking GWAS that kind of nicely illustrates the ability of genetic correlations to work through looking at both like how well your primary GWAS of a phenotype tracks with other measures of something that you think should be tightly correlated with your phenotype. So risky behavior correlated with GWAS of, say, how likely you are to speed, or your drinking behavior, or your number of sexual partners, along with looking at kind of more cognition behavior stuff that gets looked at from other things. And I talked already about the brainstorm analysis for looking at psychiatric and behavioral traits, but so kind of the updated data there strongly showing that something like years of education has strong, broad genetic correlations across the spectrum of psychiatric disorders, and even into many things that would classically be thought of as more neurological, and thus maybe more biologically driven and not as tightly related to behavior. But seeing that higher education associated with, say, lower risk of stroke, both ischemic and early onset. And so you can imagine many things you might be interested in doing with genetic correlations. So not only in terms of comparing phenotypes, but it may be interesting to find phenotypes that are easier to study than your really hard to collect phenotype, but maybe are closely related genetically. There's examples of that in the EA2 paper. You can find sets of phenotypes that might make sense to meta-analyze together because they're sufficiently similar that there's shared information between the phenotypes. So for example, this is the kind of motivation for the MTAG method, the paper that Patrick led. I think there'll be at least a brief mention of that next week. You can identify traits that have some form of G by E or G by sex, depending on how to think of things that affect sex differences. Based on genetic correlation to the same trait in separate groups of individuals. So this is similar to the idea of what might drive those cross-cohort differences, but looking at it as instead of an ascertainment artifact, whether you're interested in finding some differences there. So there's been examples where, for example, the genetic correlation of waist-hip ratio, if you do the GWAS in men versus if you do the GWAS in women, you do see RG dropping fairly substantially below 1, even if you collected people from the same cohort. And then maybe you want to take your genetic correlation and look at questions more related to statements about causality or about the specific nature of the pleiotropy. So there's a range of methods, some of which will be talked about next week. You can think about taking the genetic correlation and seeing if those same associations between risk for two phenotypes exist within families. You can look for kind of the SNP level, whether effects seem to be shared across traits. So that's a method called ASSET. There's a method that extends LD score as a moment-based method that's looking here at the cross product. It starts looking at fourth moments of the GWAS scores that might get you towards looking at directionality in these correlations. You can also model the matrix of genetic correlations in a method called Genomic SEM that will be talked about next week. Or you could start using this as the genetic correlations as fodder for what things you think might be worth looking at Mendelian randomization for. Resources on the last slide that basically say there's code available, there's resources in terms of asking questions, there's resources in terms of checking your genetic correlation across a whole range of publicly available data, and there's browsers for looking at other people's publicly available data, especially out of UK Biobank. So lots of support across the community for being able to look at these things, both in your own data and other people's data. That's all I've got. Yeah, it's definitely possible. I know a few different efforts that are working on that question in various domains. So one is the observation that doing exactly that kind of partitioning in the Gremel framework does seem to help with robustness there. So some of the results I showed a comparison LD score to other estimation methods was including various forms of Gremel that are including partitioning by MOF and by LD and showing benefits of that. There's a method called HESS that tries to look under a fixed effects model, goes kind of regionally across the genome and looks for localized correlation, and thus is able to make statements about if there are regions where there seems to be covariance versus regions where there doesn't. That model has its own quirks and idiosyncrasies, but at least is an attempt at that kind of idea of partitioning, less in terms of allele frequency, but more in terms of locations in the genome. And there's ongoing work to play with those ideas. We're just taking essentially the partitioned heritability model and applying it to genetic correlation. There ends up being some thorniness in terms of figuring out what quantity makes sense to report out of that. In terms of talking about these, because you're talking about fractions of a covariance term, and so thinking about what the natural equivalence of an enrichment or like something like a tau, those kind of things, it's not necessarily clear like what the interpretable output is there, but there are certainly ideas in that direction, and I think it's an active area of research still.
I have a question about the interpretation of genetic correlation. So is there any other way, intuitive way to interpret each kind of number, like 0.7, 0.3, for example, you can say 0.6 of genetic correlation between two things is twice as large as genetic correlation of 0.3, or something like that, or yeah, I'm just curious about how we can interpret those correlations and explain the grandma.
Yeah, it's a good question. So in the sense that we have at least a little more trust that these are unbiased estimates than, say, the SNP heritabilities are, and they are in a natural, like 0 to 1, well, negative 1 to 1 scaling, negative correlation. There is room to start making those more quantitative statements. There's not, so there's partially a question there about whether, if you want to start talking about whether one correlation is twice the other, whether you want to make that statement in terms of the correlation, or whether you want to talk about the squared correlation in terms of more the variance explained component. But I don't think there's any like general sense of like, there's not a guideline of saying that like 0.3 is a large correlation or a small correlation. It's more just, here's a correlation, interpret as you see fit. There is potentially the wrinkle there that, so unlike, say, a phenotypic correlation, the error in your measurement doesn't diminish a genetic correlation. So all measurement error will do is drive down the SNP heritability, but the genetic correlation will remain robust. And so there's not quite the same statements. It doesn't naturally translate to take, say, rules of thumb that exist in psychology or any other field about like what constitutes a large phenotypic correlation and say that should equivalently be our benchmarks in genetic correlation. So, but relative to that statement, I don't think there's a community consensus on what would be a large genetic correlation.
I think there's a paper in the 70s about how to interpret genetic correlations. I can't remember who it's by, but under some simplifying assumptions, obviously simplifying, you can interpret it directly as a proportion of overlap. Right? So 0.7 correlation is interpreted as a 70% overlap between genes, assuming the genes have exactly the same effect, and that's divided by the total. So I think it's Anson. I'll look it up in a second.
Yeah. That makes sense with some simplifying assumptions. You can make stronger statements like that. I think we're already over, so probably.